The development of induced pluripotent stem cells, also known as iPS cells, in 2011 had the promise of providing an alternative option to embryonic stem cell use. These cells are sourced from somatic cells, which are differentiated adult cells. Hi, my name is Claire Darpuche, and I'm going to discuss with you today the research of the immunogenicity of induced pluripotent stem cells by Zhao, Zhang, Rong, and Zhu. The aim of this research is to investigate the immunogenicity of induced EIBS autologous cells when introduced to B6 standard black mice. This study used B6 male standard black mice and a single line of embryonic stem cells and used the novel episomal approach of integration. Mouse embryonic fibroblasts were isolated from a B6 embryo and transfected with a P-cos LNP vector that expressed the 4 reprogramming factors. These were then grown in culture. The cells were then injected subcutaneously into the hind region of the B6 mice. Teratomas that were formed were removed. They were then fixed with a 4% formaldehyde solution or frozen. These were then stained with a hemoxantin eosin stain or with antibodies. A microassay was also taken to evaluate gene expression. So we can see in Figure 3 is a summary of teratoma formation, showing that 80% of the 64 individual mice developed teratomas. In Figure 4, we can shows a stain of evidence of tissue necrosis in the regressing tumours of the B6 mice. In Figures 5, we show T-cell infiltration we can see throughout the teratoma samples, which are indicated through the red markers. Our antibody stains that used were indicated along the top. Figure 6 is the results of our microassay, showing that 9 genes were found to be overexpressed throughout the teratomas. So what does this all mean? Investigating the gene expression in the teratomas derived from the B6 iPSC mice showed that 9 genes were commonly overexpressed, specifically the 2 known tumor-causing genes HORMAD1 and ZG16. They also found that this response was not evident in embryonic stem cells. From our results, we can conclude that the iPS cells create an immunogenic response, including teratoma formation, T-cell infiltration, and tissue necrosis. In 2013, a study which used multiple synagenic embryonic stem cell lines and different iPS cells using the same genetic sequences did not correlate with the results of the previous study. They found that although tumours and T-cell infiltration did develop using both iPSC and embryonic stem cells, the results between the two did not differ significantly in expression. We can conclude from these results between the two studies that more study is needed before iPSC cell use for medical purposes. Thank you.